In this video, we're going to be looking at how a number of environmental factors can drastically alter the ability of a protein, including enzymes, to perform their function. Now, before we actually get into the proper scientific terminology, let's look at these conditions and what actually happens to proteins under these. Now, for anyone watching the video that has eaten an egg before, they know that eating a raw egg is probably not a very good idea because there might be microorganisms inside of the egg that can make you sick. So what do you do in order to make the protein in the egg consumable? Well, you add a lot of heat. You increase the temperature, and we can see because the color of egg white actually changes. Raw egg white is actually colorless, but heated egg white, once it gets up to a certain temperature, actually turns white. Now, in chemistry, we recognize that a color change is one of the indications that a chemical change has occurred, meaning that we are actually altering the chemical structure of this protein at the molecular level. What we have just observed when we take the protein within egg white and heat it up is a phenomenon that we call denaturation. If we apply this to the protein, we say after the egg white has been cooked, the protein within it has become denatured. Now, what happens at the molecular level is that when we heat up a protein, this causes the protein structure to move faster because when you increase temperature, you increase the amount of kinetic energy every object at that temperature has, and the protein actually moves so quickly that it literally shakes its own structure apart and falls apart into basically a linear chain of amino acids, losing all of the details of its 3D structure. Now, one of the interesting things about protein denaturation is that there are actually multiple factors that can cause protein denaturation, one of which being very, very highly concentrated acid, as well as very, very concentrated base as well. So when a protein is dropped into a highly acidic or basic environment, the presence of ions in the acid will actually disrupt the forces that hold this protein together and cause it to fall apart. And if you actually have a strong enough acid and drop that strong enough acid onto an egg white, you can actually watch the egg cook in the acid in front of you. Now, because salt is also made of ions, as long as you have concentrated enough salt, meaning that you dissolve enough salt into a small enough volume of water, you can effectively get the salt to do the same thing as highly concentrated acid and base. In fact, using salt as a preservative, this is one of the reasons why salt works so well as a preservative, because high enough concentrations of salt will actually cause the proteins of microorganisms such as bacteria or fungi to denature, effectively killing the organism and preventing them from being able to consume the food that was preserved in salt. So now that we've talked about these three different factors that can cause a protein to denature, it's time to talk about what the consequences are with enzymes. Now, whether we add heat or whether we add large concentrations of acid, base, or salt, all of the complex folding that leads to a protein's 3D structure is going to be lost when the protein structure falls apart, which means the most important part of an enzyme, the active site where the substrate enters, is also going to be lost. Now, if the active site falls apart, the entire enzyme is not going to be able to function properly. This is why it's very, very important for the human body as well as the body of every organism to maintain relatively stable conditions in order to prevent the organism's enzymes from actually falling apart. And this is why the approximate temperature of the human body on average remains around 37 degrees Celsius. If this temperature gets significantly higher, usually above 40, our enzymes actually start shaking themselves apart because of the high levels of heat, and the enzymes actually lose the chemical structure that allows them to be able to function. Consequently, since acidity and alkalinity are also problems that can lead to proteins falling apart. The human body's pH remains approximately around 7 because this is a neutral pH, a pH that is right in between acidic and basic conditions, and also because 70% of the human body, approximately, is made of water, which has a neutral pH. And again, the reason for this is to minimize the amount of acid or base that is available to potentially denature our proteins and cause our enzymes to fall apart. This concludes our introductory series on background that students need in order to gain a full understanding of the digestive system. In the next series of videos, we're going to start breaking down the organs of the digestive system, starting at the very beginning, the mouth, and looking at how chemical changes as well as mechanical changes in your food done by your digestive system gradually break down the nutrients that you've learned about in this series into the monomer components and how the body absorbs those and eventually is able to use those nutrients.